coming to now. 1 year after we had published our work on the toll receptor, Charlie Janeway and Russell Legitov showed that the human homologue of Drosophila toll existed, and it could could activate adaptive immunity. Now, so we had, I'm happy to say that we had, ever from the 90s on, 95 on, we had Human Frontiers Ground together with Charlie Janeway, Russell Legitov, Alma Zinkowitz, and Fotis Kafantos, who was at that time director of the European Molecular Biology Laboratory in Heidelberg. And so we exchanged, of course, our data, and we were actively interacting, I would say. And 1 year later, 1 year after the study of Janeway, Bruce Beutler also identified a toll-like receptor gene, and he could show that this toll-like receptor gene responded to a lipopolysaccharide, which was not yet understood, which was hypothesized but not yet understood in the previous slide. Now, so we were in 98, we were in the presence, the community was in the presence of at least 2 reports showing that toll-like receptors, similar to those in the flies, could respond to the infection and activate NF-kappa B, and in particular, then activate the adaptive immune system, which is absent from the fly. Now, very rapidly then, Shizuo Akira, with namely Shizuo Akira in Osaka, it was understood, I would say at that time about a dozen of laboratories got involved in the story of toll-like receptors, and it was then understood that we humans have a dozen of toll-like receptors, mice also, it's in that range of number, and that they are the toll-like receptors in the system, as in the fly, they are transmembrane receptors, and they can sense microbial ligands in the extracellular space, and nucleotide sequences are shown here in endosomes after those have been endocytosed. Now, after sensing, so I'm not going into detail of this, the lipopolysaccharides, microplasma lipopolypeptides, and so on, also flagellum for TLR5, and after sensing those, the toll-like receptors activate via adaptor proteins, which play an important role, but again, I apologize for not going into detail of that. They will activate NF-kappa B there and lead to production of antimicrobial peptides, that was established by then, and also to IRFs, which stands for interfering regulatory factors, which will control essentially the antibiotic response. So, let me then just show one example of the structure of toll-like receptors have now been identified, established in several groups and several instances, also several species, and they are solenoid extracellular molecules with lucid-rich repeats and an intracytoplasmic domain, which is referred to as telogen. And when they bind, as illustrated here, these are double-stranded RNAs binding to a TLR3 dimer, and you see that the double-stranded RNA will bring 2 monomers together, and this bring together will then lead to also to the bring together of the intracytoplasmic signaling domains, and then the response comes. Again, the names of the people who have done these structural studies are shown here. They were not done now. So, over the years then, a surprise came to the community when it appeared that TLRs, which initially we thought, well, they had been identified as important in infection, we initially thought they might be central to infection, that might be the central role. Now, over the years, it has become apparent that surely infection is one of the roles, fighting infection or signaling infection, one of the roles of TLRs, but they also play a major role in inflammation. And you may have noticed, for those of you who have followed that type of literature, that there's not one single type of inflammation, not one single type of inflammation in clinical settings where TLRs are not involved. They also play a role in vaccination as adjuvants, as recognizing adjuvants, some of the adjuvants, not all, there's some which do not go by TLRs. They play a role in autoimmunity, they play a role in allergy, They play a role in immunotherapy, and this is becoming a very important avenue of research these days. They play a role in the central nervous system. This was really a surprise for us to see in the literature that there's not one single cell type in the brain, our brain, which does not express toll-like receptors. And the functions are still not fully understood, or they're far from being fully understood. Of course, for microglia, you could speculate that those are sort of macrophage-type molecules, and there would be a role in inflammation, but it's much more complex. And finally, they also play a role in the kidneys, important role. So there is nearly, in our own body, there's nearly no aspect which does not involve somehow toll-like receptors. And I'm told by people who are following the bibliography that there have been about the last 18 years now, about 25,000 papers on TLRs, essentially in clinical settings. And of course, we haven't time to go into this, but of course, this gives a whole variety of new possibilities for therapy, and this is really being developed in many, many clinical settings now, all over the world. Now, I must say at this stage that the initial notion which we had that TLRs were sentinels against microbes and activated by microbial ligands, I'm putting here endodomini 2000, that, and we all believed that, I mean, in the community, and we had, of course, many meetings, and we discussed all this with our colleagues on the Human Frontiers grant and later of the NIH grant. That notion turned out to be too restricted. Second point, in fact, many diverse molecules, and namely damaged self, can act as TLR agonists. This had been sort of anticipated by a person named Pauline Metzinger at Bethesda in Washington, but it had been severely disputed by many, and it has now been established. And third point, the idea that TLRs were the unique NF-kappa B activating receptors for microbial and damaged self ligands that also turned out to be too restricted. And let me say a few words about these points. Let me show you here TLR agonists in sterile inflammation. And remind you again that around 2000, we all believed in the community that TLRs receptors were there to recognize microbes. But now it has appeared that in sterile inflammation, the absence of any microbes, TLRs can be activated by debris from necrotic, injured, hypoxic, tumoral cells, extracellular membrane fragments, immune complexes, DNA chromatin complexes, and so on. And I'm giving here some examples, HMGB1 interacting with TLR2 and also TLR4, TLR9, and so on. This has essentially the experimental evidence is based on cell cultures in which TLRs have been knocked out by molecular biology methods or in mutants. Now, admittedly, there are very few studies, in contrast to what we have seen for the interaction between TLRs and microbial ligands. There are still a very few experiments or demonstrations, structural demonstration of the interaction of these molecules here with the TLRs, and in one case, for instance, HMGB1 has been shown to interact with a co-receptor of TLR4 and not directly with them. Okay, that is certainly, I would say that this is one of the important new avenues of research, and it is being addressed in many laboratories now. Now, let me then say, before going to the next picture, let me just say that we find the same in the flies. Damaged cells can also activate the immune response, and I'll come back just one word at the end of my presentation. Now, so let's now turn to the idea that TLRs, toll-like receptors, are the only only receptors recognizing infection. I said this turned out not to be true. Let me put the TLRs again here. They are membrane, transmembrane molecules, as I said. But now they're also C-type lectin receptors. They're also on the membranes. They had been reported before the toll-like receptors, but it had not been understood that they can activate NF-kappa B. This has been demonstrated in the 2000s. Now, in addition to those, there are node-like receptors discovered around 2000, And there are the RIG-I-like receptors, discovered in 2004, RLRs, and there is now C-gas. And I'll just say a very few words on these before coming to the conclusion of our presentation. Because all this is all these are new data, and it's interesting for us to see how those receptors evolve over the hundreds of millions here, which are behind us. So, also let me point out here that the number of these receptors is maybe, we are uncertain about the C-type